And then I have one that was due on Friday. I haven't finished it, but yesterday I had to put it in. And then I was gone for the Wednesday break. I don't know why, because I was here for quiz. I don't know, I just skipped that for some reason. And I didn't get the results from my lab. And then right now it's like, because I didn't. Yeah.
You can pass with
That's
you can pass even if you have to be terribly on the test.
That's what saved me in 401 last time. You know, I pray that thank you letter from that scholarship they gave me. I pray thank you letter.
I probably have to do that.
Well, it's for only a few people. And we write thank you letters every year. And I wrote it. I wish I might have to do it.
That's pretty.
When I, remember when I told you about how bad with my studies I needed? Got it tore up by the dog? No, that's it. So I started rewriting that and everything. And I was like, why? I was so pissed off. I couldn't remember anything I said. I only wrote like a page. And I had like 3.5 pages. Yeah.
How's your day going, Hannah?
Pretty good. How about yours?
Alright. First, when I came in this morning, I was not able to log into my account, my computer account, so.
Oh, that's annoying. Yeah. How was your weekend?
Well, it's fine. Got, let's see, what did I do this weekend? Went out for lunch with my wife, did a little shopping. So, how about you?
I got that much homework, so that was nice.
Oh, okay.
I didn't get a lot yet.
Nice, nice.
Last time I had my personality, it was so funny, I forgot to thank you guys.
How are you two doing? Good. So, enjoying the frosty weather? Things will change, right?
Yeah. Hoping it'll be sooner than we think. Huh? The cold weather.
The weather.
Switching back to warm.
How's your day going?
I want to go out.
I did the weather coming out. Constant changing temperatures.
Okay, you home already, or are you gonna be coming out? So when's your next game?
This Friday, I'm 64.
Oh, okay.
Really?
Well, hopefully the weather won't warm up, right?
So that means my dad definitely.
Good. So how are you two doing today?
Unless we just like, yeah, we'll drop you off. That would totally.
Done with one exam today.
Columbus doesn't.
Oh, what test was that? Oh, okay. Did you pass?
I think I passed. We'll see how See what I actually got for free, but I don't know.
But next,
What do they, there's an expression I learned from a student. It's, or some kind of like a mantra. C to degree. Have you heard that one?
no, they don't. It don't. In this field,
What do you mean by that?
in our field.
Oh, yeah, in Europe.
Yeah, and like,
In my field,
Alright, how you doing today?
I'm sick of this cold weather.
Yeah.
But I'm also sick of the changing weather, so it's like gonna be hot or cold, quick, I'm sick.
Right,
Cause I'm sensitive to like the changes, and I've been getting migraines left and right, and I'm like, why is this happening to me?
it could
All
be
people.
in Marquette.
I hope you're feeling better.
Why are in Marquette? Devin, my best friend, she goes to Northern, to Marquette. She got.
Here.
Using for your pain.
Fucking penknife. And I was like holding it, it wouldn't, I couldn't get it. And then I pushed, rotated it to the other side. I was talking to Devin, my roommate, and I was like, I would have
Yeah, great.
Good, how are you?
Good, good.
and it did stop being
What's your season?
No, I leave on Wednesday for Kentucky.
Is that for like?
Yeah, so it's semifinals, and then after that, if I qualify, then I'll go to nationals. Yeah, I'm just missing a lot of class, but it's okay.
Like 3 days.
We're leaving on Wednesday because we have to get the horses there early so that we can ride them, like get them ready. Yeah, and then, so Friday, then we'll do like pattern preview, so that's like, the people will do the pattern on the horses, and then the horses will get chosen for like which competition they'll go in. And then I'll show on Saturday, and then my teammates will show, I'm not sure when I'm showing, but Saturday and Sunday is the show, and then we'll come back Monday. So, it's an 8-hour drive, but with the horses, it'll probably be like 12 hours.
It's been a month since I bought it.
I put gas in my car last week, it was under $2, I put gas in my car.
I guess it was like maybe about 4 last day that you missed.
Yeah.
Okay.
At least I have both my labs on Tuesday, so at least I won't miss that.
Right, good point, good point.
That's how Lex did. She put
All right.
$100 in her car and she got it.
Look forward to, right?
Yeah, thank you.
I was gonna say so.
I was like, yeah, I wish.
No shame. Me, Lauren, and then.
Hey, how's it going?
I was on the back.
How are you doing?
It'll be us three.
I was just sitting there. Like studying my skirt.
One more now, how are you doing? All right, looks like you're nice.
I'll be okay, but he's like,
Hopefully, one more day's ahead, right?
I don't either.
I literally don't know. No, I literally just said, you know.
I lost my sweatpants for the day, so. I originally had pants, but.
Oh, you Megan, you got hands on?
Oh, yeah. I have my pants. Yeah.
For like lecture, and I also have to record.
Well, good afternoon, everyone. Well, we finished chapter 12 last week, and I did upload the assignments on Blackboard so that you can upload your notes for chapter 12. So, looking at the schedule, our next exam is Monday, March 30th. So we got plenty of time. So that's actually the deadline for uploading your notes here, so. Okay, so we have that on Blackboard, and let's see, this week we're gonna start learning about viruses, viroids, and prions, so that's chapter 13. For Tuesday's lab, we're gonna take a look at some of the tests that you set up last week with your bacteria. Then we're going to be able to fill out and complete that one master table. So then we can get an idea of what you've been working with all these weeks. And then we're also going to set up our next experiment with working with fungi. So we're going to transition from bacteria to fungi for a couple of weeks. And then we're going to work with the viruses after that. So this is kind of like a little preview of what's to come. Well, the main subject of this chapter is about the viruses, even though it also includes the viroids and other things there, but those are just like minor things that they're mentioned at just at the very end of the chapter. So the emphasis is on viruses. All right, let's begin with a question. Are viruses alive? Are they technically alive? No, they are not. Do viruses have cells? All right, they do not. They are not made up of cells. That's right. And yet they have, life-like properties. So when we look at a virus, we're looking at two main parts to it. First is that it contains genetic information. So we're looking at either DNA or RNA. So it's kind of interesting that some of these viruses actually use RNA, not DNA, as a means of storing information for its replication. And then DNA or RNA can be either double-stranded, like there's two chains of nucleic acids, or it could be single-stranded. So I know like when you first learn about DNA, you learn that it's double-stranded, and you learn that RNA is single-stranded. Well, viruses are unique. They can have single-stranded DNA. So what does that look like? Just one chain of nucleic acids instead of two. And there are some viruses that actually have double-stranded RNA. So, similar to the way that you can have two strands of nucleotides to make, your typical DNA double helix, you can make the same type of structure using RNA nucleotides. Okay, so we have viruses contain genetic information, and then that is surrounded by a capsule or coat made up of protein. So think of the protein coat as like the container, the outer covering that that covers the nucleic acid. And that's basically it. That's your virus right there. So it's kind of like a container that transports genetic information. And then the virus is dependent on a host cell in order to replicate. So that host could be a human cell. It could be another animal. It could be a plant cell. It could be a bacterial cell. And these viruses are super specific for their host. So in other words, typically, like a virus that affects bacteria cannot infect you. So that's the kind of specialization that we're looking at there. And then what we're going to learn is that the virus needs the enzymes and other kind of components that are inside of that host cell in order to replicate, in order to make more viral nucleic acid, whether it's DNA or RNA, and also more protein. So the ability to make that nucleic acid, make the protein, is dependent upon using the processes that are contained within the cell that it infects. All right, so that's our kind of like our introduction to viruses. Any questions so far? All right, let's move on here. I think we've seen this term before. It seems like there's a lot of information in this book that just kind of repeats itself, but that's okay because I think it's all right to hear information more than one time. So, yeah, I'm pretty sure we actually have seen this word before. It's called the bacteriophage. So that's the name of the virus, the class of viruses that infect bacteria. And that's actually what we will be working with in about 2 weeks here, the bacteriophages. All right, I think most of you know that viruses are very small. How small? So here's I added this kind of like scale here in order to give us a sense of how small these viruses are. In addition to these numbers, we also have pictures to help us with that. On the left, we have just a small sliver of what is supposed to represent a human red blood cell. So just for size comparison, this would be the outer surface of one of your cells. And then you probably recognize this bacterial shape here. What do we call this shape? Bacillus, that's right. So this is that's the kind of shape that Escherichia coli, or better known as E. coli, has, right? So we're looking at dimensions of about 1 to 3 micrometers, or microns. So from the top to the bottom of this bacterium here is about 1 micrometer or 1 micron, which is equivalent to 1,000 nanometers. So I know that the fine print can be a little bit hard to see there. So we're looking at nanometers, which are about 1 billionth of a meter. So try and envision your meter stick. Divide that into a billion lengths of equal distance, and that nanometer would be equivalent to one of those parts. All right, so then we're looking at different kinds of viruses up above. So here, this little speck here on the slide represents a poliovirus responsible for the disease we call polio. And look at that. We're looking at something that's about 30 nanometers in length. And then, they do vary in shape here. But then the largest virus over here on the right is about 800 millimeters. So, I'm sorry, 800 nanometers, I'm sorry, misspoke there. So the unit that we're focused on is the nanometer, as opposed to bacteria where the scales in the terms of micrometers. So we're that much smaller here. All right, let's look at parts. So remember that we learned that the viruses are composed of nucleic acid surrounded by protein. Okay. That makes up what's referred to as a virion. So the virion is what we call the virus. Think of it as a synonym for the virus. So again, we're talking about the protein coat plus the nucleic acid that's inside. So that's our virion. And then we have another synonym here for the protein coat, and that is the term capsid. So capsid is also refers to our outer covering made up of protein. Now that capsid is actually made up of protein subunits. So for example, in this picture here, our capsid is in orange, kind of like that orangey red there. And you see all those little circles? They're supposed to represent like spheres. Each one of those circles is supposed to represent an individual protein subunit. So you have a bunch of subunits that combine to make up the capsid. And it's hollow on the inside, so that's where the nucleic acid, which is colored green, is located. All right, what else do we have going on here? Well, we can have structures attached to that capsid that help with the attachment of that virus to its host cell. So these little items here that look like little pins, or I don't know if any of you do any sewing or not, but these are like pins here. These are called or named spikes. So these are also made of protein, but they extend off of the surface of that capsid. So when that virus comes in contact with the host cell, like my hand over here, it's the spikes that adhere to the host cell. And then in addition to that, bacteriophages can have they're kind of like landing gear, referred to as tail fibers. We'll see another picture of that coming up soon here. So I'll point that out once we get there. All right, what else do we have going on? Well, as it turns out, in addition to having nucleic acid and protein, there are some viruses that have a membrane that surrounds all that. And that's referred to as an envelope. So some viruses have an envelope, some of them don't. So this membrane is pretty similar to your standard plasma membrane. It's a lipid, phospholipid bilayer. That's what we're looking at here. It's composed of not only lipids but also proteins. So proteins are part of the membrane. And those proteins can also help with attachment to host cells. Now this is interesting here. The last point here. You remember that some viruses lack a membrane. So this is an envelope virus down here. This one is a non-enveloped, or sometimes referred to as naked virus here. But then, the point here that's made is that these viruses, the ones that don't have the envelope, are more resistant to common disinfectants. So that'd be kind of like your Lysol spray, they use for like cleaning surfaces, that kind of stuff. And it's because those disinfectants have detergents. You know, detergents that are designed to break up and solubilize lipids that you'd find in a membrane. So because this virus doesn't have that lipid, that membrane, is not, you know, impacted as similar to the way that an envelope virus would be by a detergent. All right, how are we doing so far? Still awake? Okay, barely hanging in there. All right, let's keep going. Let's look at, hey, there's some different shapes. So it's kind of, remember that when we learned about bacteria, we learned that there were some common shapes. You know, we reminded ourselves about bacillus, you know, you have coccus. You know, those are the most common ones. Then you could have spirillum, for some other spirochetes. Here we're only looking at 3 shapes, so nothing too crazy going on here. The first one, icosahedral. Okay, so it's made up of a bunch of triangles. So, like, I know it's a little bit hard to see here, but like this lighter part here is one triangle. Can you see that? I'm trying to draw it out there with my pointer. Can you see that triangle right there? Here's a triangle, one triangle. Everyone see that? Okay, there's one, but then you can see that there's other sides to this thing, and then we can have up to about 20 of them arranged in a 3-dimensional shape. So that kind of gives us our icosahedral. Well, some of you are into like playing board games, and you've probably seen some dice that are kind of like that, multi-sided dice. Yeah. Okay. I don't know, have you ever had a 20-sided die before? You ever seen one of those? Yes? Okay, that's pretty cool. All right, so that'd be the shape of the capsid. Then another shape that we see here is called helical. Helical. And I know when you look at this thing, it looks like a cylinder, like there's an open, you know, it's like shaped like an open, an empty column here. So it's named helical because of the pattern in which these subunits are arranged around that central opening. So I don't know if you're familiar with like a paper towel roll, like the inner tube of a paper towel roll. That tube is constructed in a spiral. So it's similar to that. Think like have you ever played with a spring, like a slinky when you were a kid? You remember playing with that? That's also a spiral, but when it's all together, it looks like an empty cylinder. So that's basically what we're looking at here for helical. And then inside that opening is where the nucleic acid is located. Okay. And then the last shape is called complex. It's actually a combination of icosahedral and the helixes. So that's what we see here with our bacteriophage. Okay, remember last time I mentioned that these bacteriophages can have appendages called tail fibers. So now we actually see what the tail fibers look like here. It almost looks like spider legs, right? So think of this as like the landing gear, how this bacteriophage can attach to a bacterium before infection. All right. All right, how we doing? We're doing all right? 3 shapes? Good. Taxonomy is a little bit different from what we see with living organisms. We're only looking at, a little over 10,000 different viruses. So that's kind of shocking. I thought there would be 1 million. But no, there's only about a little over 10,000. So it's not that many if you think about it. And then as far as classification schemes go, we're going to see one way we can classify these viruses, but in concept there are many more. For example, the way that I learned it was that we could classify based on types of nucleic acid, whether they have DNA or RNA. And then you could further break that down based on whether they had single-stranded or double-stranded, and then go from there. What we're going to see in the next slide here is that we can classify them, at least those that cause diseases, based on what they call their route of transmission. So in other words, how do they enter into our bodies and cause infections? So that's what we mean by a route of transmission, the path to infection. So this is another way that we can categorize viruses. So on this slide, we're actually looking at 4 different groups, based on how they can infect. So that's realizing that, but we also have to keep in mind that there's a lot of viruses that don't infect, but these would be like the medically relevant viruses. All right, let's take a look at that first group called the enteric. So they're named because of the way that that involves the human intestines. So they can spread from feces, that are released from our intestines, and then they can actually be, consumed basically, or either accidentally eaten or imbibed orally. So that's the name. That's how we have the name fecal-oral route. So here are some examples on the right. We have the enteroviruses. One example of that would be polio. So I don't know if when you're in elementary school, did you learn about the different presidents of the United States? You remember which one suffered from polio? Let's name another one. Yes. FDR. It was Franklin Delano Roosevelt. That's right. What happened there? Well, we have a virus that can exit out of the human body, in the form of feces, and then we learned on Monday that sometimes you can have wastewater contamination of various bodies of water. Like sewer systems can back up or whatever, and that and then these pathogens can enter into lakes and streams. So FDR actually swam in a contaminated lake, unintentionally, and he didn't do it because he knew it was contaminated. But and that's actually how he acquired the polio virus. So maybe unintentionally, got some of the water in his mouth, and then that entered into his body that way. So that's actually the most common way that these enteric viruses enter into our body, is if we accidentally consume or imbibe contaminated water. Okay, let's keep moving on here. Well, I should also mention that these noroviruses are also pretty common too, and a lot of times you hear about the importance of hand washing. And that's because these viruses can actually, they can literally spread out of a toilet. You probably have seen those movies or videos of those atomic bomb blasts where they can create a mushroom cloud. Well, some of these toilets, they inject so much water with such force into the toilet bowl that it will create an invisible mushroom cloud. You'll have all these very fine droplets of water that will cause and carry up bacteria and viruses out of the toilet and cause them to literally rain down on surfaces. So it could be the toilet seat, it could be the floor, it could be the counter. And then people unintentionally will touch objects that contain those pathogens and then accidentally spread them to their mouth. Okay, so that's also very common. So that's the reason why when there's these norovirus outbreaks, you have these big public health campaigns telling people to wash their hands because they may accidentally touch surfaces that the viruses have literally rained down on. Okay, I know it's kind of a weird concept, but that's the way these things can spread. All right, let's look at the next category, respiratory. So named because those are spread through inhalation. So that's true for influenza, which is the long name for the flu. So the influenza is the same thing as the flu. You may have seen pictures of people sneezing or coughing and expelling tiny droplets of mist into the air that can contain these viruses. And then people accidentally inhale that air. So like how do we prevent that? Well, that's one of the reasons why if you're ill, you should wear a mask so that you're not spreading these droplets into the air that could contain viruses. So another reason why I want to have better ventilation in this room, so that if you guys are sneezing and coughing and breathing, we can draw those things out of the room. Okay. All right, number 3 on our list are these sexually transmitted viruses. So that's exchanged as a result of intercourse. So we're talking about fluids moving between males and females. It could be through semen, for example, or vaginal fluids, that kind of stuff. So there's a number of these sexually transmitted diseases. Here are some examples. And some of them are, they could be lethal. We've seen that with the with HIV, the human immunodeficiency virus, for example. All right, and then we have our last category are the zoonotic viruses. So the zoonotic ones are the ones that are can be found from animals. For example, you could have infected. Think of it like an infected dog that accidentally that may bite you and then transmit the virus that way. Rabies can also be spread by raccoons. I mean, some of these animals are natural carriers of these diseases, and when raccoons actually defecate, the virus will actually be in their feces, and then you can actually accidentally come in contact with that and acquire rabies from that. And then we also talked about vectors. So, what do we mean by an arthropod? Or give me an example. Or just think about just an example of a vector. Maybe that might be easier for you. Like a mosquito, that's right. That could bite an infected, could be an infected person and then transmit to another person. We can also think about like Lyme disease, for example. Like what would be the vector for Lyme disease? Ticks, and ticks can feed on infected anyone? Deer. There you go. So deer would be the animal, hence the name zoonotic. And then through a tick biting the deer and then biting you, you could acquire the disease. Lyme disease is probably not a good example because that's actually bacterial, but kind of gives us same concept. Works with viruses in addition to bacteria. We have other things. We talk about the malaria already with the plasmodium. That works also with the vectors. Spiders are also vectors. Spiders. So you can actually get diseases, from a spider bite too. All right, next we're going to take a look at life cycles of these viruses using the bacteriophages as an example. So how do they infect and how do they reproduce? All right, we're looking at a couple of different terms related to these different viral states. So the first one is called lytic phages. Anyone know what they mean by virulent? Virulent. Right there.
Highly transmissible.
It's highly transmissible, very good. So it's infective. It can cause, in this case, because we're dealing with a bacteriophage, it can infect the bacterium. And then that's going to be contrasted to what's referred to as a temperate phage. So temperate phages can go into an inactive phase. Think of it as like dormancy. That's referred to as a latent state, a state of inactivity. So So we have, two different, phages named based on whether one is, highly aggressive, highly infectious, infecting lots of cells versus one that is not. Okay, so we're going to be taking a look at these two different states here in this chapter. One where the virus is rapidly replicating, producing more virions, and then spreading and infecting other cells, versus the latent state where the virus may infect a cell, but then go into a dormant, silent state, and then not cause that cell to die, but rather in this dormant state, when that host cell replicates, it is also replicated. All right, so we'll see examples of both of those coming up here. All right, did you bring your binoculars with you so that you can read this? Or do you have the eagle eyes? All right, so remember that we learned on the previous slide that there's these two types of phages. We're going to start off by looking at the first one, the lytic ones. So these are like the highly infectious ones, the virulent ones. So these are named because they will rapidly reproduce after infection, and then are released when the cell dies. We're going to take a look at these 5 steps in the subsequent slides. So when we take a look at the next 3 slides or something or so, we'll see the exact same pictures again. So don't worry if you can't record everything at this moment here. But anyways, we're going to take a look at these 5 infectious steps, basically, and the first one on the next slide will be what we call attachments. The virus is initially outside of the cell, but then will land and physically attach to a host cell. So that's attachment. And again, we're focused at this point with the bacteriophages. So, do you remember what we call this shape here? What is the shape?
Complex.
It's complex, that's right. Because we have the icosahedral attached to the helical shape below, and then we have the tail fibers. Tail fibers are kind of like they look kind of like a spider there. It helps to the virus to adhere to the outer surface of that bacterium. So that adherence is actually to receptors. So on the surface of that bacterium, there's typically proteins there that are kind of like, you've probably heard of the lock and key model. These tail fibers will fit precisely and attach to specific receptors. Maybe think of it like a jigsaw puzzle. There's a perfect fit there between the tail fibers and these receptors. Now these receptors are not there to be, their role is not to be places for sites of attachment. I mean, that's just a consequence of evolution. But they have, different, defined roles of their own that are separate from infection.
All right, so after attachment, then we have genome entry. Okay, what do they mean by genome? Anyone? Genome refers to it's a DNA or RNA, that's right. Very good. I know it looks like a gnome, but it's actually a genome. So it's DNA or RNA. And remember that's represented by the green colored string-like structure. That could be DNA or RNA. So what we see in this step here is that the virus is kind of like a miniature syringe. It's like a syringe that will inject that genetic material inside of that cell. All right, now we're up to step number 3. This one's called synthesis. So now that the genetic material has entered into the host cell, then we have transcription and translation. Okay, so who remembers? What do we mean by transcription? You're making RNA using DNA, and in this example, let's just say that, well, in this example, we're using viral DNA. So transcription is happening using the viral DNA in order to make viral RNA. So try to imagine you have viral RNA in here being synthesized. And then we have translation. And what's made by translation? You have proteins, that's right. So you're using the information in that viral RNA in order to make viral protein. And those proteins are the subunits that make up the capsid. So they're parts that make up the protein coat. They're parts that make up those tail fibers. All right, and then we also need more viral DNA. So we have DNA replication. You know, the viral DNA is used as a template to make more viral DNA. So that's what we mean by synthesis. All these things are being made here. More DNA, more proteins. So, all right, so what's happening next? Let's go on to step 4. So here's synthesis, where we have more viral nucleic acids, more viral proteins made. But then in step 4, now we see assembly. Okay, so try to imagine that before the capsid's made, initially you have all those subunits. You remember we saw all those little circles, all those red circles that make up the capsid? So those come together in order to make a hollow capsid. So that's what they mean by an empty head. And then the idea is that in the assembly process, the nucleic acid is placed inside of that capsid, and then we have attachment of the other protein parts to make the complete virus. Okay, so then step number 5, release. Okay. Looks like the cell's dying because it's rupturing open. Okay, so that's cell lysis. You know, lysis means splitting open and then release of the bacteriophages here. Okay, burst size of an example virus called T4 is approximately 200. So 200 of these bacteriophages are made, you know, from one that infected the cell. All right. Let's see here. What's the next slide here? I think we're actually gonna stop here for today. Looks like a good spot. Any more questions? All right. Well, in that case, have a great day, everyone. I'll see everyone tomorrow.